गुड मॉर्निंग डियर स्टूडेंट्स, वेलकम टू जेनेसिस। आज के इस वीडियो में हम लोग पढ़ने वाले हैं अबाउट किंगडम मोनेरा। सो पूरा वीडियो सिर्फ बैक्टीरिया को डेडिकेटेड है। लेट अस अंडरस्टैंड सम वेरी इंपॉर्टेंट कैरेक्टरिस्टिक फीचर्स ऑफ दिस किंगडम। यहाँ जो भी बैक्टीरिया हैं दे आर ऑल यूनिसेल्युलर एंड प्रोकैरियोटिक, दैट मीन्स न्यूक्लियस हैज नो न्यूक्लियर मेम्ब्रेन। सेकेंड, इनका जो न्यूक्लियस है उसमें हमें मिलेगा डीएनए, आरएनए एंड नॉन हिस्टोन प्रोटीन्स। अगर हमारे न्यूक्लियस को देखा जाए देन इट कंटेन्स डीएनए, आरएनए एंड हिस्टोन प्रोटीन। ऑन टॉप ऑफ दैट रिमेंबर one thing, इनका जो डीएनए है इट इज डबल स्ट्रैंडेड जस्ट लाइक आवर डीएनए। द डिफरेंस इज हमारा डीएनए होता है लीनियर, सो हियर आर द टू स्ट्रैंड्स, दे आर लीनियरली कॉइल्ड अराउंड ईच अदर। लेकिन इनका जो डीएनए है इट इज सर्कुलर एंड इट इज ऑल्सो स्मॉलर, वे स्मॉलर देन आवर डीएनए। सो द टॉपिक है सेल वॉल। वी नो प्लांट्स का सेल वॉल सेल्यूलोज से बना है, फंगी का सेल वॉल काइटिन से बना है, बट द सेल वॉल ऑफ अ बैक्टीरियल सेल इज मेड अप ऑफ समथिंग कॉल्ड पेप्टिडोग्लाइकन। है क्या? इट्स अ कॉम्बिनेशन ऑफ एमाइनो एसिड्स एंड शुगर। ये पेप्टिडोग्लाइकन सारे बैक्टीरिया के सेल वॉल में मिलता है? नहीं, सारे बैक्टीरिया के सेल वॉल में ये वाला पेप्टिडोग्लाइकन नहीं मिलता है। देयर आर फ्यू बैक्टीरिया जो बिना पेप्टिडोग्लाइकन वाले सेल वॉल्स हैं। दोज आर कॉल्ड आर्कीबैक्टीरिया एंड माइकोप्लाज्मा। सो यू नीड टू रिमेंबर दिस दैट बैक्टीरिया में सेल वॉल तो होता है बट नॉट नेसेसरीली ऑल ऑफ देम विल बी मेड अप ऑफ पेप्टिडोग्लाइकन। देयर आर आर्कीबैक्टीरिया एंड माइकोप्लाज्मा व्हिच आर एक्सेप्शंस। नेक्स्ट इज द मेम्ब्रेन बाउंड ऑर्गेनेल। मेम्ब्रेन बाउंड ऑर्गेनेल्स like Golgi bodies, endoplasmic reticulum, these are not seen in bacterial cell. But there is one organelle जो bacterial as well as eukaryotic cell में दिखता है and the name of that organelle is ribosome. So हम घुमा के ये कह सकते हैं कि ribosome is the only organelle which is present in a prokaryotic cell as well as in a eukaryotic cell. But दो different types के हैं। जो prokaryotic cell वाला ribosome है it is called 70S type and जो eukaryotic cell वाला ribosome है it is called 80S type. Now my question for you, अगर तुम्हें पता है what is the function of ribosome in a cell, write down in the comment box. Next is shape. Shape हमने देखा है बैक्टीरिया में mainly 4 shapes दिखते हैं. One is coccus which is spherical, bacillus rod shape, then spirillum is spiral and last vibrium comma shaped. Next is life processes. अगर इन बॉडी को देखा जाए then there are certain primary life processes which are seen. जैसे digestion हो, respiration हो, excretion हो. But when you look into a bacterial cell there are only 2 main life processes. One is respiration, another is nutrition. इस टॉपिक को हम लोग डिटेल्स में पढ़ने वाले हैं because on the basis of these 2 life processes बैक्टीरिया को कैटेगरीज में डिवाइड किया जाता है. And the last point is repetition of what we have already studied in our previous class कि ये जो किंगडम मोनेरा है इसे बाद में तोड़ दिया गया है. इसे तोड़ के 2 सब ग्रुप्स बनाए गए हैं. One is Archaebacteria, another Eubacteria, and the Eubacteria is further divided into Cyanobacteria and Bacteria. We are going to study about them in details. Now, इनके ये जो 2 लाइफ प्रोसेसेस थे, रेस्पिरेशन एंड न्यूट्रिशन, इसे जरा समझते हैं. See, रेस्पिरेशन ये दोनों टाइप के रेस्पिरेशन एग्जिबिट करते हैं. Some of them are aerobic, that means they need oxygen. Some of them are anaerobic. But apart from that, there are 2 more varieties. If an organism can only and only survive in presence of oxygen, तब हम उसे कहते हैं obligate aerobes. If they can survive only in absence of oxygen, तो हम कहते हैं obligate anaerobes. So remember one thing, obligate word is for somebody who's not at all flexible. So when you say obligate aerobes, then remember इन्हें oxygen की जरूरत है. Obligate anaerobes, they do not require oxygen. There is another terminology जहां हम facultative word use करते हैं. F for facultative, F for flexible. That means ये flexible हैं. They can survive in both the conditions. So अगर facultative aerobes हैं, that means primarily they are anaerobic. लेकिन अगर oxygen रहा तो वो मर नहीं जाएंगे. They'll be able to survive. Similarly, facultative anaerobes means they are actually aerobic, but if there is absence of oxygen, they still manage to survive. Respiration के बाद life process है nutrition. Nutrition बैक्टीरिया में primarily 2 है. One autotrophic, that means they can make their own food, another heterotrophs. Major percentage of bacteria heterotrophic ही हैं. Now under autotrophic they are again of 2 types, photoautotrophic and chemoautotrophic. अगर वो खाना खुद अपना बना रहे हैं by taking energy of light, then we call it photoautotrophic. But if they are obtaining that energy by using chemicals, especially inorganic compounds, then we call them chemoautotrophic. Heterotrophic means there are three types. Number one, saprophytic, if they are obtaining nourishment from dead remains like dead plants or dead animals. Then symbiotic, we know symbiosis is a relation where they have a relation with an organism or a host plant and where both the organisms are going to be mutually benefited. एक बहुत simple example बचपन से पढ़ा है हम लोग ने, rhizobium, which is found in the root nodules of leguminous plant. तो root nodules के leguminous plant में जब ये लोग रहते हैं, then what do? They help the plant in fixing the atmospheric nitrogen into compounds of nitrates, which help in the growth of the plant. So that is another type of heterotrophic mode of nutrition. And the last type is parasitic, where they obtain nutrients from a living host. Saprophytic means from dead remains, in parasitic they obtain it from living hosts. Now let us discuss the mode of reproduction in bacteria. हम सबको पता है that they are very fast at reproducing, and this is the reason why they still survive in this environment, right? Because they can reproduce very fast. They have two modes of reproduction. They can reproduce by asexual mode of reproduction and also by sexual mode of reproduction. As you already know, in case of asexual mode of reproduction, the body will basically split into two. There is no need of another bacteria here. So it is either by binary fission, where the body is dividing into two new cells, or sometimes when the favorable conditions are not available, in those conditions, in those situations, the bacteria within itself produces certain structures which are called endospores. These are extremely resistant structures and they remain within the cell. And when the cell bursts, these are released out in the environment. They will remain in the environment and wait for the right type of conditions to come. As the right conditions arrive, they break open that resistant wall and grow into a bacteria. So there are two methods by which under asexual reproduction they can reproduce. One is by binary fission, another is by the production of endosperm. But the difference is, when the conditions are favorable, they will choose binary fission. If the conditions are not favorable, then by endospores. Now let's discuss how sexually do these bacteria reproduce. Now in this, obviously when we are talking about sexual reproduction, we will talk about two different bacteria exchanging their genetic material. But to understand this, let us look into the structure of a bacterium. See, the bacterium has basically three protective layers outside it. Number one, the first protective layer is called a capsule. The second protective layer is the cell wall, and the third one is cell membrane. Now, inside the cell membrane, there is cytoplasm, and what is expected that in the cytoplasm there are going to be organelles, but I've already told you they do not have membrane-bound organelle. The only organelle that it has, they are known as ribosomes. So here are ribosomes which are present inside the cytoplasm. What else do we find? We find a nucleoid instead of a nucleus. Why? Because the nucleus is not surrounded by a nuclear membrane, so we call it a nucleoid. And how does the DNA look like? The DNA is circular and it is double-stranded. So here is the DNA. But can you see there is one more extra structure that I have labeled? This is known as plasmids. Now let's understand what are plasmids. These are extra chromosomal DNA. That means apart from the chromosome which is present, made out of the DNA which is present inside the cytoplasm, they also have an extra chromosomal DNA. Is it mandatory that all bacteria will have a plasmid? No, it is like that extra power some of them have. What is that extra power that they have? This, if it is present, then such bacteria, they usually become antibiotic resistant. So it's a huge power to them which helps them to survive, right? So now this plasmid, if it is not present in all the bacteria, then other bacteria would always want to have this brahmastra with them, right? So now to obtain a piece of this plasmid, the only process that they have is to perform sexual reproduction. Apart from this, I have drawn two more structures in the diagram. These are known as appendages. One is the flagellum, another known as pili. Flagellum is mainly for locomotion. Pili is again used in sexual reproduction. When, how, I'm going to explain it now. So now let's come to sexual reproduction. There are three ways by which they can perform sexual reproduction. Number one is known as transformation, number two conjugation, and number three transduction. In the first method, which is called transformation, remember here there are two bacteria which we will consider. One which wants to give its DNA, another which wants to receive. The one which gives its DNA is known as the donor, and the one which receives it is known as the recipient. But in this transformation, the donor and the recipient will not have to be in contact with each other. So the donor will basically release a piece of DNA which will be directly absorbed by the recipient. So remember, transformation is one such kind of sexual reproduction in bacteria which requires no direct contact. Second method is conjugation. Now conjugation is shown by those bacteria which contain plasmid. Okay, so this will be a transfer of plasmid between one which contains the plasmid and the other bacteria which lacks it. Now for this, the two bacteria will come in contact, will make a tube, a conjugation tube, and through this conjugation tube the plasmid will be transferred from the bacteria which has plasmid to the one which lacks it. The third type is called transduction. See, in this, for transfer of genetic material, the two bacteria, they do not come in contact, but they use somebody for this. Who are they using? They are using bacteriophage. What are bacteriophage? They are viruses. Okay, these viruses take the genetic material of one bacteria and sit on the other bacteria and help in transferring of the genetic material. So we discussed that sexual reproduction can happen with the help of transformation or by conjugation or by transduction. Now, before we understand the different modes of reproduction shown by bacteria, let us understand the structure of the bacteria. In general, the bacterial cell is covered by three envelopes. The first one is called capsule. This can be capsule or also can be a slimy layer, then we call it a slime layer. This is the outermost layer. Next to it is the cell wall, and next to the cell wall is the cell membrane. So the layer which covers the cytoplasm of a bacterial cell is the cell membrane. Now inside the cytoplasm we see there is only one kind of organelle present, which are known as ribosomes. Apart from that, in the cytoplasm there is the circular DNA. And remember one thing, the quantity of the DNA is as much as it is required to make one chromosome. So if you are ever asked that how many chromosomes are there in a bacterial cell, then in a bacterial cell there is only one chromosome. Apart from this, there is also another piece of extra chromosomal DNA which is present in bacteria, that is known as plasmid. Plasmid may be present in a bacteria, may not be present in a bacteria. Bacteria contain appendages like flagellum, which is mainly for locomotion, and some of them contain appendage which are known as pili. These structures, pili, they basically help during the process of sexual reproduction. So when we understand sexual reproduction, we will see what is the function of pili. And please also remember the term plasmid, which is the extra chromosomal DNA found in certain bacteria, because we would need these two terms when we understand sexual reproduction in bacteria. See, we all know that bacteria, they can grow very fast and they are huge in number, and this is probably the reason why they cause diseases in us and we find it so difficult to fight them. And you know the reason why that they have such huge population? Because they have both sexual and asexual mode of reproduction ready for them. When they primarily want to reproduce, they prefer taking up asexual mode of reproduction, which will be either by binary fission, where one bacterium divides into two, or by endospore formation. What is endospore? These are highly resilient structures which are formed within a bacterial cell when the situation is not at all favorable, and they wait for the right time to arrive. As soon as the favorable environmental conditions are set, they will release these endospores outside, but otherwise their mode of reproduction will always be binary fission. So the two mode of reproductions, asexual mode of reproduction, one is binary fission when situation is favorable, and if the situation is not favorable, then they choose forming endospores. Now let us talk about sexual mode of reproduction. Before I explain sexual mode of reproduction, remember this process is mainly for genetic combination. The bacteria would want to perform sexual mode of reproduction when it wants to merge its genetic material with the genetic material of another bacterium. So there are three methods by which they can do it. Number one is called transformation. See, this is a very simple concept of absorbing characteristic feature from the environment. Imagine you go to a class where in the class you find most of your friends are interested in painting, and probably you have no idea or no interest in painting. But if you are kept in a class like that for some couple of years, you will also transform yourself to some extent, where you are going to start liking, or where you are going to develop some liking towards painting. So what are you doing? You are basically absorbing the characteristic of your friends, and you are also transforming yourself. The same thing can happen even in bacterium. Say for example, a bacterium has some characteristic feature, but when it comes in contact with an environment where there are other bacteria with some other characteristic features, it will have a tendency to absorb the DNA of those bacteria if they are are given a chance, and they will try to imbibe those characteristic features in them. So imagine a condition where a non-virulent bacteria, non-virulent means something that does not cause disease, when comes in contact with, not physical contact with the environment where there are virulent bacteria, then it will have a chance to transform itself into a virulent bacteria. This is transformation. So it's basically direct absorption of genetic material from the environment. The second method is called conjugation. See, when we explain conjugation, here you have to come back to those two terms that I had explained. One is plasmid, another pili. Plasmid, we know it is an extra chromosomal DNA, so it is not available to all bacteria. There are certain bacteria which can contain these, and those who have this, they are more powerful because they get some extra powers. What are those extra powers? They are resistant to antibiotics, they are more adaptable to harsh environmental conditions. So those bacteria who actually thrive in very harsh environmental conditions are usually found to contain plasmid, whereas the bacteria which survive in a more congenial environment, they lack plasmid. But those who lack will always have a tendency to take plasmid from the powerful ones. And how can they do that? Imagine one bacteria with a plasmid apart from their DNA. This will be known as the donor, and there is another bacterium with its own DNA but no extra chromosomal plasmid. So now when this has to transfer its plasmid to this is known as the recipient. There will be formation of a tube through which they can be transferred. So it will make a copy of the plasmid. So there will be one copy of the plasmid, and this plasmid will be sent to the next bacterium. But to do that, you need a tube. That means we basically want the two bacteria to come in contact with each other and hold each other. To hold themselves, they require these structures which are called pili. It is usually shown by the bacteria that contain the plasmid. So with the help of its pili, it's going to hold the recipient bacteria and then transfer the plasmid. This method of sexual reproduction is known as conjugation. And the third method is called transduction. In transduction, there will be no direct absorption of DNA like it was in transformation. There will be no direct contact of two bacterium to transfer genetic material like it was in conjugation. But here we are using somebody for transferring genetic material from one bacterium to another. And this is a bacteriophage. What is a bacteriophage? A bacteriophage is a virus which basically infects bacteria. So when this bacteriophage sits on one bacterium and then from one it goes to another bacterium, it takes the genetic material of the previous bacterium and injects it into the second bacterium. See, if you want to understand this, this is very similar to what is done by bees when they sit on one flower, and from one flower when they move to another flower, what are they doing? They're basically transferring the pollen grain. So similarly, the bacteriophages, they transfer genetic material of one bacterium to another bacterium, and this process is known as transduction. Now let us discuss about Archaebacteria. We all know that kingdom Monera is divided again into group Archaebacteria and Eubacteria. Archaebacteria का मतलब है those who are primitive. So this basically comprises of primitive prokaryotes. And remember one thing, these bacteria are found in extremely harsh environmental conditions. Second point is their cell wall lacks peptidoglycan. This is where Archaebacteria differs from Eubacteria. And this is the reason why we basically had to divide kingdom Monera into these two separate groups. Archaebacteria का जो cell wall है, it is not made up of peptidoglycan. Instead, it is made up of branched lipids. And ये जो branch chain lipids हैं, they actually make them very tough and resistant. And this is the reason why they are found in harsh environmental conditions. There are three types of Archaebacteria. सबसे पहला type है methanogens. These are those bacteria who can convert carbon dioxide into methane. ये usually they are found in the gut of ruminating organisms, okay, like buffalo and other different kinds of cattle. And they are the reason why we use dung to produce biogas. You know, in biogas plants, we use dung which gets converted to release methane. And these microorganisms which are present in the dung of the ruminating animals, they convert carbon dioxide directly into methane. So they are of great economical importance to us. The second one are halophiles. Halophiles means they are capable of surviving in extremely salt-rich environment. And the third category, thermoacidophiles. They are resistant to two parameters. They are resistant to extremely high temperature as well as extremely acidic environment. They are mostly found in hot sulfur springs. Understand Eubacteria. Well, under Eubacteria, there are many subgroups, but as per our syllabus, we are only going to study Cyanobacteria and Mycoplasma. But before that, let us have a general understanding of them. Eubacteria, definitely in their cell wall, they have peptidoglycan. Now, there is a process of staining bacteria which is very common, and it is known as gram staining method. While this method was performed on eubacteria, it was found that they are again of two types. Those who retain the color of the stain, they are called gram positive bacteria, and those who do not retain the color of the stain, they are called gram negative bacteria. Usually it is found that the gram negative bacteria, they are little more virulent, means disease causing, compared to gram positive bacteria. Now let us find out what is this difference that is causing one bacteria to retain the stain, whereas the other bacteria loses the stain. See, in case of gram positive bacteria, the cell wall has peptidoglycan plus something else which is known as teichoic acid. This teichoic acid is absent in case of gram negative bacteria, so they do not have it. Second, they retain the gram stain because of very thick layer of peptidoglycan, which in case of gram negative bacteria is thin. On top of that, the cell wall of the gram negative bacteria has very high lipid content, and because of this high fat content on the surface of the cell wall, they never can retain the color slips away. For gram positive bacteria, a very common one is E. coli, and the example of a gram negative bacteria is Bacillus subtilis. Now let us understand cyanobacteria. They are unicellular. They can exist in colony or in the form of filaments. Okay. Remember one thing, their body, if it is in the form of colony or in the form of a filament, it is usually covered by a gelatinous sheath. In this diagram, I have tried to show you how different cells, they arrange themselves, and this becomes one structure because it is covered by this mucilaginous sheath. Then they are photosynthetic in nature, and the pigment that they have is chlorophyll A. Some of them can actually fix atmospheric nitrogen in some specialized cells that are with them, those which are called heterocysts. Now here in this structure you can see there are many cells, but I have shaded two cells here just to show you that these are those specialized cells that can fix atmospheric nitrogen. Examples of nitrogen fixing cyanobacteria are nostoc and Anabaena. And one last very intriguing feature about cyanobacteria is they lack flagellum throughout their life cycle. That means they do not have that appendage for locomotion in their body. Now finally let us talk about mycoplasma. ये बहुत ही अजीब टाइप के बैक्टीरिया हैं, and they are very interesting as well. See, they are non-motile. They cannot move from one place to another because they do not have flagellum. Second important point, they are pleomorphic. That means they can constantly change their shape. इसीलिए इन्हें jokers of plant kingdom के नाम से भी जाना जाता था. Next is they are the smallest monerans. इनसे छोटा और कोई बैक्टीरिया है नहीं. Next is they lack cell wall. इनके पास cell wall नहीं होता है. That is why they are pleomorphic. They can constantly change their shape. Now this is a very big advantage to them. You know, there are certain antibiotics जिन्हें prepare ही किया गया था to break the cell wall of a bacteria, because we knew if we can break the cell wall of a bacteria, it will be very easy to attack and kill them. But इनके पास cell wall ना होने के कारण they are resistant to all those antibiotics which attack the bacterial cell wall. उसका एक example है penicillin. So they are resistant to cell wall attacking antibiotics like penicillin. Next is इन्हें हम PPLO के नाम से भी जानते हैं, which is pleuropneumonia like organism. You know why? Because they were first discovered in the pleural fluid of cattle. Pleural fluid क्या है? We all know lungs के outside membrane को कहा जाता है pleura, and between the two pleura there is a fluid which is known as pleural fluid, and they basically infect that pleural fluid. So with this we have finished kingdom Monera. I know there's a lot that we have discussed, so unless and until you revise, read it, it's going to be very difficult for you to remember. Now soon we are going to start the next kingdom. Post your doubts, share the channel with your friends, and keep revising. Thank you.